Hello class 10. Last day we were talking about tonicity and the three types of tonicity. Now today we'll start with the proper explanation of the three types of tonicity. Now isotonic means, iso means same, that means the relative concentration of water molecules and the solute on either side of the cell membrane is the same. In such a solution, there is no net movement of water molecules across the cell membrane. That means no osmosis will occur. Clear? Next is hypotonic. In this condition, the solution outside the cell has a lower solute concentration than the fluids inside the cell. As a result, the water molecules from outside will move into the cell. That means end osmosis. That means just now, in the last class we have read about end osmosis, and we know that end osmosis will only occur when the concentration of the solute will be lower outside the cell. That means the water is more outside. So water will move from its higher concentration to its lower concentration. That means from outside to inside. Hypertonic is just the opposite case. In this condition, the solution outside the cell has a higher solute concentration than the fluids inside the cell. Consequently, the water molecules from the interior of the cell will move out. That means outside the cell, the water concentration is low and solute concentration is high. So the water concentration is more inside the cell. That means the water will go from inside to outside the cell. Okay? So that is hypertonic. The three terms isotonic, hypotonic and hypertonic are also expressed as isoosmotic, hypoosmotic and hyperosmotic. Okay. Now what is the result? The cell shape and size remain unchanged. The solution outside is isotonic. Cell shrinks in size and loses shape. The solution outside is hypertonic. And the cell slightly enlarges or even bursts. The solution outside is hypotonic. So these are the three results we can find from here. If it is isotonic, then the cell shape and size will remain unchanged. If it is hypertonic, then the cell will shrink in size and loses shape. And if it is the solution outside is hypotonic, then the water will get inside through end osmosis. That means the cell will enlarge. Now students, you see this diagram. This diagram is there in your book also. Animal cell and plant cell. They have shown that what is happening in isotonic solution, what is happening in hypertonic solution, and what is happening in hypotonic solution. So these three are very, very important, and maybe that you are given the diagrams and you have to answer the questions. So these three diagrams are important. As you can see, when it is hypertonic outside, the water is coming out from the cell and the cell is shrinking. This one, number B. This one. Okay. Now another phenomenon is active transport. Now what is it? It is the passage of a substance from its lower to higher concentration using energy from the cell through a living cell membrane. So here the most important word in this diagram is this one. Using energy from the cell. So active transport is the passage of substance. We all know that generally the substances move from its higher to lower concentration. But here you see it is different. It is moving from its lower to higher concentration. So definitely it needs an external source of energy. So that is why it is known as active transport. Certain nutrients such as ions of nitrates, sulfates, potassium, zinc, manganese cannot pass through the cell membrane of the root cells easily. This is on account of one main reason, that their concentration is higher inside the root cells, and it is so maintained in order to develop osmotic pressure for absorbing water. In this way, the concentration gradient of these ions is opposite to that of diffusion, because we know that diffusion always will occur from higher to lower concentration. Now students, this difference is very very important for your boards. Difference between diffusion and osmosis. They have given 5 points. All the 5 points are important, and even they have differentiated the definitions. So you have to write 5 points in your exams. Now if they have given 6 marks and you have to write 6 points, then you can include the definition part also. Okay. Now when we are talking about energy, we all know that this energy is nothing but the ATP. So by using ATP, which is available as the chemical form of energy in biological systems. Clear? Now, there are three terms which you need to know. That is turgidity, plasmolysis and flaccidity. Now, what is it? When a cell reaches a state where it cannot accommodate any more water, that is, it is fully distended. Fully distended means swollen up. It is called turgid. And what is the condition called as? Condition is called as turgidity. Clear? So, the state is turgid and the condition is turgidity. When a cell is turgid, its wall is stretched under pressure from inside, and in its turn, it presses the cell contents towards the center of the cell. The pressure of the cell contents on the cell wall is called the turgor pressure, and the pressure exerted by the cell wall on the cell content is called wall pressure. So there are two types of pressure. One is turgor pressure, another is wall pressure. So the one which is exerted by the cell contents on the cell wall, that is turgor, and the one that is exerted by the cell wall on the cell content is the wall pressure. When turgid, the cell is in a somewhat balanced state. No more water is entering or leaving it. Its turgor pressure counterbalances the wall pressure, and therefore there is no further absorption of water, even though the concentration of solutes inside the cell may be greater than that outside the cell. So that is, you can say, one of the advantages of the turgid condition, because even if the concentration is not proper inside, there will be no more water entering or leaving when it is in a fully distended form. Now this diagram is important. As you can see, the turgor pressure, the wall pressure, the osmotic pressure, they are showing every kind of pressure with the help of the arrows, and it is represented together in a single diagram. So this diagram is important. Plasmolysis and flaccidity. Now what is this plasmolysis? See, these three definitions are important. Plasmolysis is the phenomenon of the contraction of cytoplasm from the cell wall caused due to withdrawal of water when placed in a hypertonic solution. Actually, plasmolysis means the shrinkage of protoplasm, or the shrinkage of, you can say also, the cytoplasm. But for a proper definition, you need to learn this. But you have to understand the definition that it is nothing but the shrinkage of the protoplasm or cytoplasm. And when this plasmolysis is occurring, the condition is known as flaccidity. It is the condition in which the cell content is shrunken and the cell is no more tight. The cell is then said to be flaccid. We have learned turgidity and turgid. The same thing, the opposite one is the flaccidity or flaccid. And turgidity we have already read about. Okay. Now, uses of turgidity to plants. Turgidity provides rigidity to soft tissues such as the leaves. That we know because it is important for the mechanical support and the stiffness of the plant, right? Turgor pressure helps to push through the hard ground, as in mushrooms and in a seedling. Sometimes the roots of the certain trees have been seen to crack the walls or a concrete floor of an adjoining building. This again is due to turgor pressure. So turgor pressure is actually, it helps to push through the hard ground. Because the roots are going inside the soil with the help of this turgor pressure, because if the roots are not turgid, then it cannot push through the hard soil or ground. Turgor in root cells builds up root pressure, and this upward flow of water is due to a heavy pressure from the roots, which is called root pressure. Which water? The water which is getting inside through the root hairs, and it is moving or traveling through the stem up to the leaves. So this whole movement, we all know that it occurs through the xylem tissues. So that movement of the water is possible only because of the tightness of the adjacent cells. And what are those cells? Those are root cells, and it is brought about by the high root pressure. Here. So now we need to know the proper definition of root pressure. This is the proper definition of root pressure. It is the pressure developed in the roots due to continued inward movement of water through cell-to-cell osmosis, which helps in the ascent of cell sap upward through the stem. So this definition is important. You have to learn it. Loss of cell sap through a cut stem is called bleeding. Just you need to know what is bleeding, that's it. Turgor in the opening and closing of stomata. This though we have learned already, right? Because in the photosynthesis chapter, we were learning about the opening and closing of stomata, and where there we have seen that when the photosynthesis is occurring, when the glucose is being synthesized, then what was happening? It was making the Gerd cells turgid, and in turn this Gerd cells turgidity was helping to open the stomata. And when at night there is no photosynthesis, that means no synthesizing of glucose, that means no turgidity, it is shrinking and it is closing the stomata. Another one is turgor movement. Now turgor movement, I hope you all have heard about Mimosa pudica. So it's a very sensitive plant. Drooping of the leaves of that Mimosa pudica or touch-me-not plant, if one of the leaves is touched even lightly, the leaflets fold up, and within 2 to 3 seconds the entire leaf droops. If the leaf is touched somewhat strongly, the wave of folding and drooping spreads from the stimulated leaf to all neighboring leaves. Slowly the leaves recover and again stand erect. In this plant, the stimulus of touch leads to loss of turgor at the base of the leaflets and at the base of the petioles, called pulvinus. So this is one of the, is the best example of turgor pressure we can find in touch-me-not plant. Okay students, now this diagram is very important. As you can see, it's a cross section of a root showing cell-to-cell conduction of water from root hair to xylem. So this is very important. Please go through this diagram. It will never come for your drawing, but you have to practice it, or rather you have to observe it properly for the labelings. Okay students, so today up to this. Please go through the chapter. I hope there should not be any problem. And if you have any problem, you can ask me. So please go through the chapter and learn the definitions. And I think the best way to learn it is to write it down and practice it. Okay? Thank you.